choose the molecule that we have downloaded. So it is in my download folder. So here is my receptor, that is 1C9U. I will just open here. So you can see the protein receptor here, as there are some red dots in it that are basically water molecules, and we will see that there are some other natural ligands that are present in this receptor here, you can see. So we need to remove them for refining of this protein and purification of this protein. So for this purpose, first of all, I will go to this option, scripts. In scripts option, I will go to selection and I will click on select water molecules. When I will click here, so all the water molecules will be highlighted in yellow color. That means these molecules are currently selected in my system. So I will just click here on delete button to remove them all. As you can see, the yellow molecules are selected molecules and that are basically water molecules. So I will click this button, that is for delete, or we can also press the del button from our keyboard. So I'm just clicking here and all the water molecules have been removed. And on the other side, now I need to remove this ligand that is naturally present in this protein receptor. So I will again go to scripts. I will click on selection and I will select this option, select ligands. Again, it will be highlighted in yellow color. So I will just click delete button and it will be deleted from my protein. So now this is the pure form of my protein that can be used for interaction studies. So I will just again save it in the PDB format. For this purpose, I will just go to files option, save as, and I will just change the save as type option to protein databank files. And I am just renaming it as protein. So I am just going to desktop and I will save it here. So this is my verified protein receptor. And now next step is for ligand. So I will close this and now I will again go to file, open, and I will just go to downloads again and I will see where my downloaded receptor is present, that was downloaded in SDF format. So let me see where it is. So here it is in the name of conformer 3D. So I will just drag it and open here. As you can see, here is our receptor. So sorry, here is our ligand, that is amoxicillin drug, but it is in purified form. There are no impurities can be seen in this structure. So there is just one concern, that it is in SDF format, and for Autodoc Vina we need to convert it in the PDB format. So I will just go to file, save as, and I will change its file type to protein databank files, and I will change its name to ligand. So I will again save it on my desktop. So it has been saved here. Now we have two purified molecules. One is our receptor and the other one is basically our ligand. So here you can see protein and ligand. So these are basically our purified molecules. Now next step is about Autodoc Vina. So if you have any query or concern from this session, you can just ask me about the purification of protein and receptor. Okay, means all clear. So we can move to Autodoc Vina now. So I will just open my Autodoc tools from here. Okay, it will take some time to load, and then we can proceed with it. So after opening the Autodoc Vina, the first step will be about reading the receptor molecule first. So first of all, we need to input our receptor molecule. So we keep in mind that we will just input the purified molecule. So I will just go to Files, Read Molecule, and we will move to the desktop and select our molecule that is named as protein. So it is a refined molecule. I will just click here. So here the structure you are seeing is basically our purified receptor molecule that was 1C9U, and now we just need to introduce some charge on it. So we will add polar hydrogens to make it more reactive to interact with the ligand. So for this purpose, I will go to this edit option. I will select hydrogens, and then we will click on this add button. So from here, we will select polar only, and we will click OK. So when we will click OK, so you can see some charges has been enlightened up on this receptor. So basically these are polar hydrogens that have been added to this structure on the partial side. So it, it may become more positive. So after that, the next step is making as a macromolecule in the Autodoc Vena. So for this purpose, we will go to this grid option. We will click on this macromolecule, and then we will move to the choose button. So after choosing here, it will ask that select a molecule. So we will just select protein from here, and we will select molecule. So now it will save it as PDBQT file. So don't worry about these warnings. It is just saying that no bonded atoms in this and that. So that is totally fine. We will click OK. And now it is asking about the location where we want to save. So we are just going to save it in C drive Vina folder. So we will open C drive and move to Vina folder. Here I will just save it. Okay, so it has been saved in the C drive Vina folder. Now the next step is about grid settings. So as we know, the grid is basically a representation of a box that actually tells the Autodoc Vina where might be our active sites are present. So it actually automatically detects the active sites, and then we may just target our ligand here. So I will just go to grid again. I will click on the grid box, and then I can see a box here that is actually representing the position of active sites. So uh, minor modifications are required here, so I will just reduce the dimensions. From here we can normally set it to the 26, 30, or 32 roundabout here, so I am just making it 30 for all of them. So all the dimensions have been adjusted to 30, and then here is spacing. So basically spacing is the area. So I prefer to target the area, more and more area to get the more accurate results from here. So I will just make it to maximum, that is 1.0, and we will not touch these values. So we need to note them down, or we can just save them from here on files, output grid dimensions, and then I can just save it on my desktop, so my readings will be there. So it has been saved. Now after adjusting all these parameters, I will just go to files and close saving current. So it will lock this area for interaction. So I am just clicking to save. Okay, so my grid has been set, but it has been disappeared from here. That doesn't mean that the grid is not present here. It means that the area has been locked for the interaction. So that was the area that has been selected for this interaction. So Now the next step is to introduce our ligand in this window. So for this purpose, I will move to the ligand option. I will go to input and then open. So keep in mind that again we will just use the purified ligand file here. So it has been saved on my desktop. So I will move to desktop and I will find out my file from here. It was named as ligand. So I will just search it out, and I will find out my ligand file. So here it is, ligand. So I will just click open. I will click OK. So the ligand has been added to my molecule. So now we can proceed for its interaction. But before that, we need to set a torsion angle. Basically, torsion angle will be set to the maximum numeric value. So basically, that is the actual sides of torsion that acts with the receptor molecules. So I will go to ligand again. I will go to torsion tree and then select set number of torsions. So it has been set to the maximum number, that is 7, and then I will click on the dismiss option. So we started from the purification of protein. We purified our receptor and then we purified our ligand. Then finally we added our receptor into the autodoc vena. We added hydrogen charges on it. Then we adjusted the molecule as a macromolecule format in pdbqt and saved it in the autodoc vena folder in c drive. Then we set a grid on it and we adjusted the values of grid, and then we introduce the ligand in this file. After introducing the ligand, we set the number of torsions to the maximum. Now the last step is about the saving this ligand file into the pdbqt file to C drive Vena again. So I will go to ligand, output, and save as pdbqt. So I will click here. It will ask me that where I want to save. So we have decided that we will save it to the C drive Vena folder. So I will just move there and save my file here in the Vena folder. So my file has been saved here. Now I will just close my Autodoc window and I will move to the Vena folder again. So the Autodoc Vena has been closed now. So let's move to that folder of C drive that is known as Vena folder. So here it is present. Now we need to adjust our configuration file according to our requirements that we have done in the grid box. So we will open this conf file. We will confirm the names of our files. So our protein is named as protein.pdbqt. Our ligand is named as ligand.pdbqt. Now here we need to adjust our values. So our center values were we have saved in our desktop. So let's see what were these values so we can interpret them. So first value was minus 3.798. So I will just copy and paste it in the X. The second value was about 41.727. So I will copy it and paste again in the configuration file. Then the third value will be about Z, so that is 23.659. So I will copy it and save it here in the Z option. After that, the size values as we have adjusted it was about 30, so we will set all of them to the 30, 30, and 30. We will not touch the exhaustiveness as it is the default value as 8. So I will just click Control S, save this file, and I will close it. So now the final stage is about command prompt, where we need to use some Python codes just to run this interaction. So I will just click Windows R and then I will type CMD. So it will open a black window that is known as command prompt. So here I have to give some instructions in the Python code here to access these files. So I will just click type cd dot dot. I will click OK, enter. Again I will type cd dot dot and then I will click enter. Now I have to tell it that what is the name of folder where my files have been saved. So it was present in C drive and it was named as Vina. So I will click cd space Vina. Now, uh, now I have to enter some other commands just to access my files that are present in the cd Vina folder. So I will just type here Vina space config conf.txt. So I am telling it that my file values are saved in the configuration file that is present in Vina folder and it is named as conf.txt. Now I want to get an output in log file, so that should be named as log.txt. I will press enter here, so it will just analyze the binding sites and it will run the analysis in a few seconds. So here you can see a table that has been formed after refinement of this interaction, and nine basically values are given here. So basically nine are poses that has been used for the interaction and the best one will be given on the top rank number one. So basically this is binding affinity. So binding affinity is basically the energy that is basically absorbed when the bond formation has been taken place in this interaction. So the maximum binding affinity in negative will be considered as the best value. So this you can see this is minus 6.2 and here is the RMSD, that is root mean square density. So it should be 0 or nearest to 0. So here we can see our best binding affinity in kilocalories per mole is given as minus 6.2. So this table will also be saved in the Vina folder that we have access. So we can just open that Vina folder and we can see this log file. So we can open it and we can see the same table is just given here. So we can use it further for our different analysis. So that was about the molecular docking. So if you have any question or query regarding this, you are welcome to ask. Yes, Simrose, you can ask. Yes, the log file is automatically created in the Vina folder, but we need to make the configuration file just once. When we are going to perform docking first time, then we need to make this configuration file. So I can just share this configuration file with you, so you can just copy and paste it in the Vina folder. And every time when you proceed for the docking, just edit that file, change the values, and just proceed. Any other question? Sure, I'm just sharing after the session. Okay, so I'm waiting for the 5 minutes. If anyone has any questions, so you can ask, then we may leave. Thank you. No, Simrose, you are not audible. You can type here if your microphone is problematic. Okay, Naima, if output file shows error or could not open output file, so that means you made some mistake when creating the PDBQT file. So just review it and proceed again. Then you may fix that problem. Yes, Itama, we just performed the addition of polar hydrogens in Autodoc V. Now you will just go to the edit option, select hydrogens, and then you will select the polar only option to add these on the files. Okay, I-TASSER, okay, I will give. Simrose number of torsions, they can be set as default, but try to make them maximum when you just click that arrow present here, so you can set them to the maximum. So maximum is the best. Energy minimization means purification free. As we perform the purification in Discovery Studio, we remove all the impurities from the ligand. So that is basically energy minimization step. Simrose, if you are not able to find Vina folder, yes, basically you need to copy Vina folder from your installation folder, and then you will paste it here. Just open C drive, open program files, and you can search the Scripps Research Institute named folder. Open it. There will be a folder named as Vina in there. Just cut it and paste in the C drive. Go to program files. So there will be a folder named as the Scripps Research Institute. So open it and there will be a folder named as Vina. Just cut it and paste it into the C drive. Both tools Freya can be used, but best one is Autodoc Vina, and secondly I recommend CBDog because it also uses the same algorithms that are basically used by Autodoc Vina. So Autodoc Vina is the most authentic tool for the docking studies for enzyme and inhibitor docking. So just go with manual. If you cannot do manual, you can go with the CBDog. That is most recommended. Naima, if protein has exit diffraction of 3 angstrom and error value of 79, so that is the only option available. What should we do? So you can refine it using Galaxy Refine as we have studied it. You can just submit it in Galaxy Refine and you can do its refinement to make it more precise and accurate. Thank you, Friha. Okay, Mariam. So I can just give you a very brief overview of I-TASER. Okay. So we will just move to the results directly, and I can explain the results of it in a brief manner. So So, okay, here you can see the results page of the I-TASER. So, first of all, in the first segment, you can see the submitted sequence in fast A format. So, basically, this is the same sequence that you have been submitted to the I-TASER for the protein model. And in the second section, you can see predicted secondary structure. So, it basically tells us about the presence of helix, strands, and coils in your protein. So, here you can see under each amino acid, you can see C, S, and H is written. So basically, C indicates for coils, S for strands, and H for helices. And under this, you can see confidence score. So basically, it tells us about the confidence of that specific prediction. So that is basically 0 to 9. After that, you can see predicted solvent accessibility. So solvent accessibility is basically the solvent nature of that protein, that how much it is soluble. So you can see its value range from 0 to 9. So here you can see under each amino acid, the value ranges from 0 to 9. So 9 means highly exposed residues that are present outside the protein and that basically get the first interaction with the solvent. So 0 means buried residues that are present inside the protein. Then here is predicted normalized beta factor. So basically the normalized beta factor is the thermal mobility of each residue. So means that when heat is applied, so how these amino acids behave in the response to that heat. So basically, at high temperatures, how the vibrational motion is represented for these amino acids. So here you can see a graph that indicates the normalized beta factor from -2 to +5, and under this you can see the number of residues. So you can just interpret it. After that, here are top 10 threading templates used by I-TASER. So basically, which templates have been used by I-TASER to predict this model. So here you can see all of these templates basically taken from protein data bank to structure this protein on base of these models. After that, you can see models, top five final models predicted by I-TASER. So basically, here it gives you the five models that have been predicted by I-TASER. These are just not loaded due to, I think, internet issue. Its score basically ranges from -2 to -5 to +2. So basically, we just need to go with higher score, and basically the best model is always the model 1. So here you can see its score is -0.93. So that is basically closer to 2 plus 2, so we can use it. And furthermore, you can scroll down, you can see the different analogs. It's cofactor and coach, basically telling us about the active sites of this protein. And after that, it tells us about the gene ontology, that is basically functioning of that gene. So that was about a brief explanation of I-TASER. Okay, Freeha is asking script folder. There is no Vina folder. No, that is not possible. That if it is not there, means yeah, you have not installed the Autodoc Vina properly. So you need to reinstall it. Then you will find the Vina folder here, and then you can move it to the C drive. Okay, so any other concern? Yes, I can share link, or you can Google, so it will just give you the link to download that software. Can we go with sys modeling for refinement or protein structure prediction? Yes, sys modeling basically it do not perform the refinement. It actually predicts the structure. So you can go with it. Will it be okay for protein having? Yes, it is okay. Yes, you can use that. Is totally up to you. That is not a problem. Simrose, because I had same problem with script folder. Okay, I'm sharing you the link to download Autodoc Vina in your group. I have shared two links in your group, so you need to install both of them to properly run Autodoc Vina. One is MGL tools and another one is Autodoc Vina. So you need to install both of them if you want to perform the docking. And I have also shared the CONF folder in your group. Okay, so I think we may leave now. And if you have further queries, you can just drop your text in the group. Thank you.